Good morning, everyone, and welcome to our second edition of the Roadmap to Wellness Vasculitis webinar series. My name is George Casey. I'm on the board of the directors for the Vasculitis Foundation as the Vice President of Resource Development. Joining us today and presenting is Dr. Paul Monach from the Boston University Vasculitis Center, where he serves as the principal investigator in the Vasculitis Clinical Research Consortium. The VCRC is comprised of several vasculitis centers around North America and is dedicated to conducting clinical research in vasculitis. Dr. Monach sees patients with all forms of vasculitis, as well as general rheumatology patients in an outpatient rheumatology clinic. His specific research interests in vasculitis concern biomarkers and genetics, and he is also on the board of directors for the Vasculitis Foundation. So, Dr. Monach, please proceed with your presentation.
Okay, thanks very much, George, and thank you all who are listening. It's a great opportunity for me to try to educate as many patients as possible at one time. I've really enjoyed my association with the VF, as I expect you can imagine. So I do have some disclosures. I think it's important to know, since I'll be talking about some of these drugs, that I do have relationships in industry-sponsored clinical trials. That being said, I'll move on to the research. So I think these are some long words. They'll be familiar to some of you and not to others, but I'll come back to them because there's really no way around using the terminology. So for starters, epidemiology is essentially who gets a disease and why. Classification is a bit different, a little bit more like being a librarian, but it is important. You know, are we really, all the different names that we have for these diseases, are we really identifying the right things, or are we missing something? Pathophysiology is a long word for basically saying what is the mechanism of disease, from start to finish. What is the cause, and then how does it play itself out in terms of damaging organs? Translational research is kind of a wastebasket term for taking studies of the basic science studies into the causes of disease and trying to turn them into something that is useful clinically. I think something a little bit different than treatment trials. It's the point at which you sort of do the handoff from the lab to the people who really want to turn things into treatments, into diagnostic tests. Outcomes measures, similar idea to epidemiology, but it's the other side of it. What happens to patients after they get the disease? And how can we affect that in a positive way? And then, of course, treatment. So I'll talk about all of these. So for starters, in epidemiology, some of the main things we look at are how common are these diseases, that's the prevalence, what ages, sex, ethnicity, and what other risk factors are there. I can't go into these in detail. There's an enormous number of studies over a long period of time and in multiple parts of the world. But all I can say is that recently the development of large medical databases has been particularly useful because these are rare diseases, and so having a good quality database with, say, 4 million people in it actually enables you to look at a few thousand patients with vasculitis. I'm going to focus on genetics, not because that's necessarily the most important thing about the risk of vasculitis, and you'll see that I stressed that a couple of times, but just because that's where there's been the main area of advance recently, and that's in part due to improvements in technology for looking at genetics, and also because of really, really nice collaborations among people around the world, because to do genetic studies you actually need to recruit a lot of patients. And genetic studies do have the potential to give insights into the pathophysiology or the causes of disease, but I have to forewarn you that we're not there yet. The studies of genetics and vasculitis haven't given us clues that can necessarily be followed, and that's even true for some of the more common rheumatic diseases as well. And then finally, I have to stress the overall contribution of genetics to the risk of getting vasculitis is quite small. So a little bit about genetics in immunologic diseases. There's a region on chromosome 6 called the HLA. These were initially described as the immune response genes, which won some people a Nobel Prize back in the early 1960s. And they're an extremely variable part of our genome. And the reason is that they're associated with risk of infection. It's thought that diversity kind of helps the population cope with an epidemic infection. And so different versions of the different HLA proteins are made by different people somewhat at random, and they are often highly associated with different immunological diseases. But because they're all fairly common, and we're talking about rare diseases, the great majority of people who have the risk allele, whether it's for rheumatoid arthritis or GPA or eGPA, Churg-Strauss, don't actually have the disease. It's just a relative risk. And until recently, this was really the only known genetic risk of the different forms of vasculitis. So in addition to that, there's really been a revolution in studies of genetics recently is that there are small contributions to risk made by many common variants. And what that means is that at a given site on a chromosome in a particular gene, there may be 15% of people have one base of the DNA. It's hard to give a talk like this without going into the really basic stuff about the structure of DNA and of proteins. But I hope you'll take my word for it that there's one letter that might be in 15%, and then the other 85% of people have the other one. And so the different genetic variations that we all have are most of them are actually like that. That we don't just have random mutations all over the place. We have some of those, but there are common variants, and that's what can easily be studied. And the technology is there to actually look at every single common variant in the genome. But what that misses is the last point here, and I might as well use my little pointer if you can see it. What you don't see in these studies of common variants is what about the rare and unique variants that might be particular to an individual or to a family. And so you get that by actually sequencing the whole genome, which remains extraordinarily expensive, or what's called the whole exome, which is the part of the DNA that actually is transcribed into protein, which means that there are differences in the products. And I want to point out here that an example of the latter is a VF grant to Sharon Chung. You'll see I'll reference grants to the VF throughout this presentation, but believe it or not, the presentation in terms of the themes of different things I'm going to talk about is actually not biased. I'm not biasing that toward talking about VF contributions. We really have, as I discovered, covered elements of the important areas in vasculitis. So genome-wide association studies have been done in several vasculitic diseases. I'm not going to talk about the Behçet syndrome, which was the one that was actually done first. There again wasn't necessarily insight into what caused the disease, but it was still a nice study. One that's gotten a lot more press recently covered both GPA and MPA together. We also, through the VF, funded another one, and I was involved in one that specifically was GPA. And then also a big collaborative study in Takayasu's arteritis, also supported by the VF. And so here are some of the results. This can be a little bit daunting, but I'm going to sort of guide you through what these pictures mean. So this is first of all to say, without going into the actual numbers, that there's a strong association between GPA or Wegener's and antibodies to proteinase 3 or PR3. Similarly, there's a pretty strong association between microscopic polyangiitis, so it doesn't have the granulomatous features here, and a different ANCA antibody to myeloperoxidase or MPO. But it's not perfect. This is a complicated Venn diagram that shows there are some people who kind of cross over. Their ANCA doesn't quite match what we call the clinical phenotype. And what happened in this study was very interesting, that they found that if they split the two diseases, they either said GPA versus MPA or they said PR3 versus MPO, that really there was no such thing genetically as ANCA-associated vasculitis. What it really meant was, do you make antibodies to PR3 or do you make antibodies to MPO? And those had different genetic associations. So what this is, forget about that minus log 10 p-value. That basically says how strong is the genetic association with this. And take my word for it, this is quite strong. But again, we're strong in terms of statistical significance, the likelihood of it being true, but we're still talking about maybe a 3-fold increase. So, 1 in 10,000 chance instead of 1 in 30,000. It's not the absolute risk of getting a disease is not that high. And then the MPO is different. And so these are both in the HLA region that I told you about before that determines risk of different autoimmune diseases. But it kind of confirmed something that wasn't too surprising but had never been shown before, that a different autoantigen, meaning our own protein that's being attacked by the immune system, is different for these two different autoantigens that are components of white blood cells. But it didn't correlate so well with the GPA versus MPA, so we don't know why those two diseases are different. This was a similar study that I was involved in, led by Kathy Siminovich in Toronto, that also fine mapped, meaning I told you the HLA region is extraordinarily complicated, and came to the conclusion of a very particular gene in that region associated with the GPA phenotype. We weren't able to look at the PR3 versus MPA, but subsequently the group in Europe that did the first study actually found the same thing when they fine mapped there. So that's true, and that's kind of nice to know. It's in the HLA-DP, which was also confirming an older finding, I believe, first found in Germany, but we really found out specifically what it was. So in addition, are there other genes involved? Well, the paper from the European group also found that proteinase 3 itself, the target of autoimmunity, PR3, is a risk factor. This was known. This again was a known finding that a gene for a protein called alpha-1 antitrypsin, this is its gene name, is called serpin-A1, is a risk factor, but they didn't find anything else. And so these are things actually directly related to this protein. So we didn't get a whole lot of additional insight into GPA. This is all sort of related to the PR3 PR3 target. Why? Because this is the gene itself, and this is actually a protein that sticks to that protein in people. It regulates what it does. What we found in our study, again, we don't know why, we don't know the significance of this yet, was that there was another gene that came up as being associated with GPA. And again, we can't sort out whether that has anything to do with PR3 or not. And the thing about genetic studies, they do require very large numbers because you're doing a whole lot of different genes at the same time. The more that is done, you can sort of add the data together and learn more from them. But that's all work that's in progress. So this is a result of a genome-wide association study in Takayasu's, and here again you see the HLA region that I keep talking about. Again, this was already known, but this is confirmed in a larger study as being the most important risk factor in Takayasu's. These things are called Manhattan plots. In our diseases, they often end up being what I like to joke as is a Dubai plot. It's got one giant building in it and not a whole lot else. But there were a few other things that came up, which again will have to be investigated to see whether why they might be contributing to disease. So the interesting thing here is that these are genes that might have something to do with autoantibodies, and this is not a disease that we think of as involving antibodies at all, not a disease we would think would respond to B-cell-directed therapy such as Rituximab. It ought to be all about T cells and macrophages and granulomatous inflammation, but yet this requires some explanation. On the other hand, this gene here, this is for a cytokine or sort of an immune system hormone that is involved in activation of T cells in that pathway, so that wouldn't be too surprising. But this is all sort of, this is good quality work, but it will yield insights into the causes of these diseases, but we don't know. It'll be a while before we know. So now moving on to the classification of diseases. Some of you might be aware we've already renamed a couple of them, which can be confusing to patients, although the names are well chosen and they were done for good reasons. We really need to know in the bigger picture, do we really know what these diseases are? And there has been an advance recently, actually, just establishing that there probably are, that central nervous system vasculitis in children is probably more than one thing. In adults, it's probably more than one thing. This is a very difficult disease to study, and there are only a few centers that have enough, see enough patients and have enough pathology tissues to go through, but there are groups that are highly dedicated to doing that. But there's a big international project, which if I can get down here to, it's called DCVAS, which again is partially funded by the VF and probably wouldn't have happened without that funding, is designed to try to revise the criteria. And so it's been more than 20 years since the American College of Rheumatology did that, and there's a lot more knowns, for instance, the ANCA antibodies, but a lot more advances in imaging techniques, just better quality of data collection, more collaboration internationally, and we hope that this will either tell us that indeed polyarteritis nodosa is what it is and it's one disease, or do we have to really think of it as multiple different things with different causes. So a little bit about pathophysiology, and then I'll remind you what translational research is. So the study of pathophysiology, meaning the underlying causes of disease and vasculitis, usually means we're studying tissues or we're studying cells in the laboratory. And there are tons of different things you can do, and this is happening all the time, so I can't, I can answer individual questions, hopefully, but can't go into this in great detail. You can find a lot of abnormalities. Knowing which ones are the most important is hard, and that's why basic science research takes a very long time to sort out. But another thing that's important is the development of animal models, and there haven't been a lot of those in vasculitis, and I'll mention how that's growing and how they're starting to be used. But in any case, not everything in basic science immediately translates into, or even eventually translates into something that's useful to patients, but it sometimes does, and there's the potential for laboratory science to eventually lead to diagnostic tests or other useful clinical tests to see how a patient is doing, and to new treatments. And that whole process is called translational research. And I'll give a couple of examples that are not current, but going back. This is how the ANCA antibodies were discovered. That eventually also led to the use of B-cell directed therapies. This is how hepatitis C virus was found to be associated with the majority of cases of cryoglobulinemic vasculitis. Those are two of the biggest advances in rheumatology or in immune-mediated diseases in the past 20 years. So a little bit about study of cells and tissues recently. They do have the prospects for improving treatment, and we're actually seeing that. I'm focusing on giant cell arteritis here because I think it's where there's a big need, and there in terms of improving clinical diagnosis and treatment. And we're actually seeing some ideas becoming reality, whether they're right or not. So looking at the blood of patients and looking at temporal artery biopsies has implicated another cytokine or immune system hormone called interleukin-6 or IL-6 as being important, and that has led to the clinical trial involving an antibody that blocks that pathway and is actually FDA approved for rheumatoid arthritis. So this is a big international trial that's in progress. We don't know if it's going to work or not, but the trial is big. It's being done in a placebo-controlled way, and it'll tell us. It won't be the last study. You then have to go on to the best one and find the best way to use it if it's positive. Another thing has been the study. It would take me a long time to explain what kinases are, but these are a type of enzyme that's in the cell that they go through a cascade of activation and deactivation that lead to telling a cell essentially what to do. And in this case, these kinases are very important in turning inflammatory cells on or off, so that's pretty key to autoimmune disease, but also fighting infection. And one example, so intracellular kinases, a lot of drug companies are developing drugs that target specific ones for a variety of reasons, and so there's prospect for improving treatment eventually, but those trials aren't in progress yet. There is also a big need for improving the diagnostic accuracy of temporal artery biopsies. So we know that they're not perfect. How imperfect they are is a matter of argument. There are a lot of people who would say that we only catch 2/3 of temporal arteritis or giant cell arteritis with the biopsies because I know this patient has disease. Other people would say, well, you're treating people who don't actually have GCA. So we really need better ways to look at the temporal artery. And a couple of those, this is something a lot of investigators are looking at, and two of the most interesting ones are actually supported by grants from the VF. One is to look at a particular kind of kinase, as I told you about, what's called abbreviated as the ROC kinase, and another is a protein called 1433. I'm not sure how it got that name, and surprisingly, the antibodies have been found to it. And this is by Dr. Chakravarty and colleagues at the Cleveland Clinic. So the prospect that temporal artery biopsies, even if they don't show inflammation, could actually be stained, meaning you look for elevated levels of these proteins, and we could tell whether the patient has temporal arteritis or not, would be a big advance. So a little bit about animal models. This is not brand new, but all of this we're really talking about within the last 5 to 10 years. Previously, the only animal model of vasculitis that I know of is based on a viral infection, which still has some usefulness and some really nice studies around it, but its relevance to human diseases is harder to justify. So the biggest one was probably being able to generate antibodies to myeloperoxidase or MPO, and then to show that just injection of those antibodies into healthy mice caused kidney disease that it looks identical to what happens in patients with MPA. Now, not everything else happens to these mice, and it's been much harder to use anti-proteinase 3 or PR3 antibodies to do the same thing. So that we don't have the sort of proof that PR3 does the same thing, although I think it's very likely. But that helps. That, again, argues that, yes, these antibodies aren't just a marker of the disease. They're actually causing something, and getting rid of them is likely to be helpful. Study of this model, though, interestingly, has implicated a group of a cascade of inflammatory proteins called complement, and a particular one is called C5, and it breaks off a little piece of it called C5a. And researchers at University of North Carolina found that in this very nice model that they developed, that if they block C5a, that they actually block the kidney disease, which was somewhat surprising because that's something that usually only happens when antibodies and their target antigens like MPO are deposited somewhere rather than just sticking to a white blood cell, which is thought to be the main way that these antibodies work. And it's an idea for a new therapy that's actually in clinical trial. I know that they've successfully used it in animals. I don't know if there have already been small clinical studies, but very exciting advance to do something very different that might treat this disease in humans. A second is completely different disease, giant cell arteritis, that pieces of human temporal artery, which are obtained fairly routinely on temporal artery biopsies, can be actually put under the skin of mice, incredibly enough. This is pioneered by Connie Wand, who's one of the leaders in this field. And that can then be used to test whether new therapies can be effective. And one of the things that she's done, I mentioned the kinase inhibition as a potential future therapy. Well, at least this can actually be tested in mice first. And no mouse model is perfect, but it's better than having no model at all. Of course, we still need new models, and actually one that the VF started funding recently is an accidental finding that a basic science immunologist found where they knocked out a couple of related genes in mice, and the mice got very ill, and they have a severe form of vasculitis. This is probably not going to be a great model for any particular disease that we have, but I think it will lead to insights either into the way blood vessels are damaged or the way blood vessel damage affects tissues, or something about the diseases that actually affect people. So moving on to translational research, this is really the main thing that I do, and I'll explain a little bit what biomarkers are. So this is a fancy word for something I think that is actually familiar to any of you. Anytime you get a blood test done, that's really a biomarker of something, of whether your liver is functioning properly, your kidneys, etc. And we're looking for better blood tests to take better care of patients, and that's what that really comes down to is that the ANCA levels, and then in all forms of vasculitis, the SED rate, the CRP, level of anemia, those don't really tell us how sick somebody is, especially after they've been treated. And this is a hard thing to study, I can assure you, because we have very high standards for how well a test has to separate a sick person from a well person. I'll give you some examples of it. But among the many, many studies that are being done, there are a couple of very cutting-edge ones looking at some recently discovered things in basic biology. One is called microparticles, which are little pieces of cells that actually break off, and it's not just garbage that floats around in the blood. They actually do things, amazingly enough. So that's being looked at in patients with GPA or Wegener's. And in addition, this was a recent finding that led to a Nobel Prize just in terms of basic biology, what's called microRNA. This is RNA that is not then subsequently used to make protein, but actually regulates other RNA. These have been looked at in a number of different diseases, and so that's looked at now by someone that we're supporting to see whether that's going to provide insight into activity of GPA. And then a second thing, I don't have time to there's an immense literature here about imaging techniques, particularly in the large vessel forms of vasculitis. And so already in some parts of the world, actually, they would say that ultrasound is really helpful above and beyond the temporal artery biopsy in diagnosing giant cell arteritis. The ability of doctors to use this technique is improving. It will eventually become standard. The problem with ultrasound in general is it's not objective. It's dependent on the skill of the examiner, but you could say the same thing about how well a doctor asks patient questions or how well they examine them. So it will spread. But in the meantime, trying to really maximize the value of this is important, and we're supporting a doctor at University of Pittsburgh to look at a particular aspect of ultrasound in giant cell arteritis. Another thing that's being looked at in parallel, very different type of imaging technique, more objective but also a lot more expensive, PET scanning and then MRI in the large vessel forms of disease, giant cell and Takayasu. And these could be complementary because, for example, you can't use ultrasound to look at the aorta, the large artery that comes right out of the heart, but you could use these other techniques to do that. You can't use PET scan to look at the small arteries in the head, which you clearly need something for that for giant cell. So this is, as I said, this is my thing. So I'm not going to talk in great detail specifically about results, but I want to tell you what we need in terms of better blood and urine tests for vasculitis. We need to be able to compare them to other disease test states to make the diagnosis in the first place. ANCA is pretty good. Cryoglobulins are are pretty good. Very high eosinophils for eGPA are pretty good, but we don't have anything really that diagnoses polyarteritis nodosa. We need better stuff for large vessel vasculitis. Staging is really important. I mean, which organs are currently involved? Separating active vasculitis from a completely healthy person is easy. Separating it out from an infection, someone we know has the disease and they're coming in sick, that's hard. Separating out mild disease, I don't know if some of you have actually been in this situation, is my vasculitis just at the beginning of acting up, or is it just a cold? Is it just some very minor other condition? And then prognosis. We would love to be able to tell our patients what's going to happen to them and be able to change treatment, maybe a very small change in treatment to prevent the need for a very big treatment later. To know early on, when the patient's still very sick, are they responding to treatment? So that's why I say that which drug are we going to choose? That's the pre-treatment prediction. Should this patient get rituximab? Should that patient get cyclophosphamide? For this one, is methotrexate good enough? That kind of thing. That's a hard problem, but it's something that we can at least we can easily generate the data to look at it. Is the patient getting better, as I mentioned before? What's going to happen to them in the future? Are they going to have relapses? Are they going to be in remission? Therefore, do they need to stay on medications or not? And then other long-term outcomes, which I'm not going to go to in detail. But like I said, almost all the studies, including the ones I've done so far, we compare severe active vasculitis to patients who are in remission. That's not the need, but everything has to pass that test before it can really go on for the harder questions. So a little bit about outcomes research. What that means is we need to have long-term follow-up, and obviously this is something that improves over time because you follow people for longer, but it really requires having things in computerized databases and having collaborative groups. And so this is really accelerated in both Europe and the U.S. In the past 10 to 15 years. And so some examples, when I say we, I mean people around the world. Multiple groups have found that the MPO versus PR3 specificity for ANCA tells you a lot about whether a patient's going to have relapse or not. The two things we know that are a problem in terms of how you're going to do, especially early on with this disease, or how old you are and how bad your kidneys are. This is why we as doctors get so upset about patients with kidney dysfunction, even though you as a patient might not notice it and are more troubled by the prednisone side effects. This is a really important one, that how the choice of initial treatment of vasculitis affects the relapse rates in the long term. And really, this has been looked at again in GPA and MPA. And one thing that was discovered that oral cyclophosphamide is actually better than IV cyclophosphamide. So on the basis of one very nice trial, most of the European groups started using IV cyclophosphamide to induce remission. This is before rituximab was developed. And then when they followed these patients for longer, they found that this group had actually a lower relapse rate. So that's important to do, and it needs to be done, I think, as we start getting better treatments for diseases like giant cell arteritis, Takayasu's. And again, I want to focus on the fact that collaboration among centers has made all this work possible, or at least a lot better. And then I do want to spend, I think, more time on treatment, partly because there's been big advances, but also this is of great interest to patients. We actually do have a lot of clinical trials going on, considering how rare these diseases are. People in multiple countries have been able to get grants, and there's recently been interest from industry to actually sponsor studies, which is pretty exciting. There are multiple forms of vasculitis being studied. Not every one, I have to admit. The least common, not so much, but we're getting there. And in a couple of cases, actually, the standard of care has been changed already. So for treatment of GPA and MPA, the big finding is rituximab is as effective. This is an antibody that gets rid of B lymphocytes. So those are the cells that make antibodies. It's pretty much a sledgehammer, although not as much as cyclophosphamide, which is essentially chemotherapy. It's still an immune suppressive drug, still has risk of infection, but it is lacking in some of the other side effects. So knowing that it's as effective as this more old-fashioned drug was a nice advance. But some other things that are in progress are, is it also better at maintaining remission? It's important to know that this drug is sometimes used chronically in rheumatoid arthritis. So we know that unlike cyclophosphamide you can't take forever, rituximab you can. And whether that's a good idea depends on the individual patient. But so knowing how good it is at maintaining remission is important. And this is a study that hasn't been published yet in France, but it's been presented multiple times at some of our meetings, which indicate it is better. This is a trial that I'm involved in that has been started recently, where we hope to address the same question in a slightly different way. Another thing that I at least am excited about, this is based on a pilot study I was involved in, is a drug called abatacept or Orencia. It works by preventing the activation of T cells, so a different kind of immune cell. And we looked at it in patients with granulomatous disease because it's more less likely to be caused by antibodies. So these were chosen deliberately: B cells and antibodies, T cells and granulomas, because T cells activate the macrophages, another type of white blood cell that cause granulomas. And we had a little pilot study of 20 patients, which was encouraging. It's just been published, and so we're hoping to do a much larger randomized trial to see whether this drug really works. Sorry about that. For the patients with the most severe disease, again, sorry, that's supposed to be Pexavas. This is patients with ANCA antibodies only. We don't think it'll work in diseases where antibodies aren't really involved, but plasmapheresis is being studied in a very, very large international trial, but we've recruited more than half of the patients needed. And at the same time, addressing something that's sort of the lost stepchild in all of this is, what should we be doing with prednisone? Everyone does things slightly differently. We know it's not optimal. It probably depends a little bit on the individual patients, but two different initial regimens are being compared in this study as well. So we're not only doing the plasmapheresis aspect, which literally is like medieval bleeding with very high technology. You take the patient's blood out, you put some protein back in that doesn't have ANCA antibodies in it, and we're seeing if after the first week or so, if you can really just get half as much prednisone and do just fine. The other thing, really important study to be done that I'm involved in, as simple as it is, it's never been looked at, just leaving somebody on 5 milligrams of prednisone versus having them come off it about 6 months after they start treatment. Is that going to actually make a difference in preventing flares? And then another big trial in Europe, as you can imagine, it takes a while to do because it's the length of treatment. That's a trial in progress, but we hope to actually have the answer in a year. If you just routinely put somebody on maintenance treatment for 5 years, is that better than just doing a year and a half, which is kind of our standard? Okay, so this is follow-up of what we call the RAVE trial, Rituximab and ANCA-associated vasculitis trial, which showed that it was the same as cyclophosphamide. All this means is who's remaining in remission. So 40% of patients are flaring at some point, many of them mild flares, but some severe. And it didn't differ in the two groups. One group got one course of treatment with rituximab, another got cyclophosphamide followed by azathioprine, and they did the same. So that's why it was published in the New England Journal. But here's an example of something I gave you before where the long-term outcomes help, that the patients with MPO antibodies had a much lower flare rate than the patients with a PR3 antibodies. And this is something that's also been seen in follow-up of some European trials as well. So this has the potential to really affect decisions you make with patients. A little bit about treatment with giant cell arteritis and Takayasu. Unfortunately, this is a shorter trial because as of yet, nothing has been found to allow lower use of prednisone in giant cell arteritis. But we have new drugs that are being tried about which we're optimistic. One is a trial I'm involved in of abatacept. Again, it blocks the activation of T cells, but is widely used in rheumatoid arthritis. And we finished enrollment of this trial, and we'll finish it in a year. It's a blinded trial, so we have no idea whether the drug worked or not, but that's the way it should be. And then it'll take a while to analyze the data, but we'll at least know something. This is more designed to see, does this drug allow patients to remain in remission after they've been tapered off of prednisone in about 6 months? This is a different one. It blocks, I mentioned before, but certainly wouldn't expect you to remember, it blocks a inflammatory cytokine called interleukin-6. And so this is up and running, an industry-sponsored trial in multiple sites, I think, internationally. Takayasu is much tougher to study. Where the same trial as is done up here is actually the first randomized trial on Takayasu's. There are a lot of small series, generally done by experts around the world. And some of the drugs that have been looked at recently, which are interesting, are mycophenolate or Cellcept, which is widely used in lupus, but not so much in vasculitis, and again, tocilizumab. But I have to stress that whenever somebody publishes 10 or 20, a series of 10 or 20 patients who did well, that's encouraging, but it really certainly doesn't tell you which drug is better, but it doesn't even really tell you how well it works. So another of the big advances in treatment of vasculitis is in cryoglobulinemic vasculitis. And so for starters, probably 70% of patients overall have hepatitis C virus infection, and if their vasculitis isn't that bad, then antiviral therapy alone works. Antiviral therapy itself has actually undergone a revolution, and so even though this hasn't been studied in vasculitis, it is likely to be spectacularly successful. So the antiviral therapy for hepatitis C, traditionally with a drug called interferon, was not very well tolerated. It made people very sick. They would still do it. But much better tolerated drugs that may actually cure people rather than at a higher rate are probably going to do the same thing with vasculitis. But in patients who have severe vasculitis at the time, you can't wait for the antiviral therapy to work, and it's been shown recently in several studies that rituximab is really better than the other drugs that have traditionally been used. Unfortunately, we don't have that much evidence that it is better than any other drugs, or even how well the various drugs work in the sizable number of patients who have cryoglobulinemic vasculitis and don't have hepatitis C. They have Sjogren's, or rheumatoid arthritis, or lupus, or something else we can't even detect. These are just a couple of examples. I'm certainly not going to go through all the numbers in this table about rituximab working in cryovasculitis. It should be apparent to you, even if you're not used to looking at charts and graphs and so forth, without even looking at the numbers, that people here remained on rituximab. It isn't so scientific, but they remained on rituximab. They came off of, whether it was cyclophosphamide, azathioprine, et cetera. That's a big difference. And in another study, this is just BVAS, just means this is the activity of their vasculitis as assessed by an expert doctor in the condition. I think in this case it was mostly Carol Langford, who's now at Cleveland Clinic. And they basically, the patients who got the usual suspects, I don't know if it was cyclophosphamide or azathioprine, it was based on the severity of the disease, and they kind of did the same for 6 months. They really didn't get better, and those who got rituximab did quite a bit better. Unfortunately, the patients who got rituximab all had flares later, but this was very specifically a group of patients who couldn't get antiviral therapy, or back in those days they tried antiviral therapy and it didn't work. And so, as I said, today the options of antiviral therapy are much more effective. Finally, amazingly, there is a randomized trial already in progress for Churg-Strauss, and so I'm going to be involved in this trial soon. It's already been up and running in Europe, and this is based on an antibody to another cytokine called interleukin-5, which is particularly important in the biology of eosinophils, which is the unique feature of GPA. And there have been some very small studies which are very encouraging, so we're enthusiastic about potentially having another option. It will be a long time before we can first of all, a long time before we know that this drug works, and if it does, then to use it. But it's really, since so many patients with GPA end up dependent on some dose of prednisone, it'll be nice to have something potentially available. And this is a drug also that is likely to be very effective in this disease, but probably not in the others. So we have to be thinking, are we getting more to disease-specific things in our treatments rather than some one-size-fits-all? So finally, I don't have to go through all of this. This is my the various people that I work with. It is a highly collaborative group. The VCRC includes not only these investigators at some of the main referral centers, but very importantly, this is actually critical to getting the grant funding and to get the whole process to actually work properly to be able to do the research, is the VF and other patient advocacy groups associated with particular diseases. These are the people I work with most closely at Boston University, and these are my other key colleagues, all of whom were formerly at Boston University and now are at other places. And then finally I want to leave that up.
So thank you very much, Dr. Monach. That was excellent. I think it's exciting to see the kind of volume of activity going on. I know personally it makes me proud to be a member of the Vasculitis Foundation when I see the amount of research that the Vasculitis Foundation has been able to contribute to. I think, for those who are wondering, as a group, the Vasculitis Foundation has funded almost $2 million in research on the different vasculitides in 33 different studies, and it still remains one of the kind of 3 pillars of our mission, along with awareness and support. So I think at this point what we'd like to do is kind of have a call for questions. If people have questions around kind of the research, what's being studied, or how this can impact treatment or diagnosis, etc. For Dr. Monach, my advice is to try to keep it generic and appropriate and not get to specific patient cases. And while people are typing that, we'll give a kind of a 4-minute break, and we'll show the vasculitis awareness video, which was released this month as part of Vasculitis Awareness Month. and that will give you a chance to type in your questions. And if you don't have questions, you can enjoy the short video. So I'm going to switch the screen. I'm going to show my screen. And I'm on the vasculitis homepage right now. And what you'll find is right there on the vasculitis homepage is a link to the awareness video, which will launch the YouTube video. And I'll be quiet and let us listen and watch while you type your questions.
It's like somebody threw gasoline on me and set me on fire and said, go jump into some more gas. That's what I call the shower.
I think the low point I have 3 grandchildren, now a 4th since then. I was taking my whole family away last summer and just not having the energy to participate with them.
The diagnosis itself was a shock that I had never heard of anything like vasculitis before.
All the doctors stared at me. They were so scared, and they were frightened of me, and they didn't know what was wrong with me.
And I felt alone, and I felt scared, and I thought I was going to die before I could see my son grow.
Even though vasculitis is a worldwide problem and a global problem, we have this marvelous group called the Vasculitis Foundation that is also global and reaches out to all these different countries and is trying to provide education and support for more vasculitis research and for more vasculitis awareness among physicians.
2 years ago, I got to know about this Vasculitis Foundation in the US. And I was pleasantly surprised that the foundation exists for such rare disease. And it's a very good platform, in fact, a great platform where people who have this rare disease, they come, they meet, they share their stories.
I just remember walking into the room and seeing 100 to 300 people, all of whom had heard of the disease, first of all, and then within that had it or had relatives or friends with it.
To me, it's like being in one big family. And nobody's going to judge you because we all have some form of vasculitis.
As a nurse and a son with this chronic illness, I just feel so passionate about bringing awareness to the world, to North America.
Okay, so that's the vasculitis awareness video. You can find that again on the vasculitis homepage. At this point, I'm going to start, we'll go through some questions. We have a few minutes, and some great questions are coming in. So one of the questions I think is perfectly appropriate, Dr. Monach, and it's patients asking if they want to be involved and if they want to participate in this research, what's the best way for them to do that?
Okay, no, that is a great question. It's something we always really appreciate because sometimes that research will benefit you directly, but a lot of the times it won't, and yet people just would still do it out of the goodness of their heart and wanting to help others. So it depends where you live and what you've got. So patients should really only be in clinical trials if they're able to go to a center that's participating in it on a regular basis. That's usually how it's done. There's some we'd love to be able to enroll patients appropriately in trials from a distance, but that's rarely the case. And you have to be sick. So I tell patients all the time, fortunately, you're not eligible to be in a clinical trial. We only want to try new drugs or new approaches and people who really need that consideration. The other ways to participate, there are the VCRC, which I'm a part of. The major component of it is to be seen at one of the centers that we have scattered around the country, not covering the West Coast or the South nearly as well as the East Coast and the North. But if people are able to come in once a quarter and are able to actually travel when they're sick to be seen, that's very helpful for us both in terms of collecting long-term clinical data for the outcomes that I mentioned to you, but also potentially helping with biomarkers. And then if people are able to only be seen once at those centers, then just providing a DNA sample for genetic studies is really helpful because genetic studies require enormous numbers of patients. So if you are unable to do that, periodically we do stuff often in collaboration with the VF to try to get the word out to do online studies. And so that's something that we've done a couple of them, but would like to be able to do more through the VCRC. So those are the different ways that people can participate depending on how far away they are and kind of what their disease status is.
I think that's great. And I would like to add a plug. One of the ways the Vasculitis Foundation is participating is with a federally funded grant from PCORI, which is the Patient-Centered Outcomes Research Institute. And we're creating a vasculitis patient-powered research network with the idea of creating a large database repository of our patients, their conditions, their electronic health records, et cetera, all anonymized, aggregate, and secure, but with the ability to kind of ask questions over a larger cohort or a larger group of people. And I think that's exciting and interesting, so look for more information through the Vasculitis Foundation on that. So one of the things you just mentioned, I think that, again, is really interesting, and it seems there's been lots of development, is around genetics and understanding their role in both the disease and how it's treated. So what are there anything patients can do? There's been this advent of gene sequencing that people can, in essence, do on their own. Is that of any use or value, or is it something that needs to be done in a more controlled setting?
Yeah, I think getting your genome sequenced, I think, is not advisable. It's just might cause some anxiety and uncertainty that you can't really interpret. So it actually requires a tremendously sophisticated statistical analysis to even sort out what is the random variation that just happens to anybody and what could actually be related to disease. And the only way you get that is by comparing large numbers of people. So it's not ready for prime time, and I'm not sure that it ever will be. You have to really, sequencing will only be helpful once the actual disease-causing genes are identified, and the only way the disease-causing genes can be identified is by looking at a large number of patients, regardless of what the method used is.
I think that's a great answer, and I think that's appropriate. We don't need anything else to scare us as patients. So another kind of common theme in several of the questions is around, I think, what we would refer to as comorbidities, or people who might have other conditions that have been diagnosed alongside vasculitis, like lupus or fibromyalgia, et cetera. Is there any research being done into the fact of that, is this actually existing? Is this an error in diagnosis or a challenge in diagnosis around how these diseases either coexist, contribute to each other, et cetera?
Yeah. So that's different in very different settings. So definitely patients with lupus can get vasculitis. Usually it's episodes. We see things in rheumatology that don't quite fit the textbook. So occasionally we see somebody who seems to have lupus and then truly gets ANCA-associated vasculitis. But it's more common to just have vasculitis periodically as part of your lupus. Some patients get misdiagnosed. Sometimes doctors don't realize that inflammatory it's usually not rheumatologists, but don't realize that inflammatory arthritis can be part of the disease, and so they say they have rheumatoid arthritis and vasculitis, when you probably could consider it one thing. Fibromyalgia is an interesting one. It is not, in general, an autoimmune or inflammatory disease. However, it is at increased, I don't know if this has been looked at in vasculitis, but it's definitely patients with lupus, with Sjogren's syndrome, even with rheumatoid arthritis, have a higher rate of it, and why that is not clear. In most cases, it's not directly due to inflammation because it doesn't get better with treatment, but occasionally it does. So patients with microscopic polyangiitis or MPA, that sometimes can be the feature, just widespread pain, and then lo and behold, the reason is inflammatory disease. But the way you can tell the difference is that if it actually dramatically gets better with prednisone. So if it doesn't, then maybe indeed you have both fibromyalgia and vasculitis, and they need to be treated differently. It doesn't mean that more immune suppressive treatment is needed. It just means that they need to be approached in different ways as two separate conditions, but one gives the risk of fibromyalgia.
Okay, I think the biomarkers research is really interesting, and especially for those of us who like kind of hard data or something that's specific and measurable. And so there's a question around, do you envision a time in where you can use biomarkers as an indicator of flare as opposed to just patient symptoms?
Yes, so I do. I certainly hope so, because this is what I've done research on. It's just a long road, because at some point you have to question whether the doctor's right or not, and then show that your set of blood tests is more accurate in terms of determining whether a flare is happening. It really should be the case. I mean, we should be able to find markers in the bloodstream that are better able to tell us what's going on at the microscopic level, and there's a history of that in long-term in medicine, that getting pieces of tissue and looking under the microscope revolutionized what people could diagnose 100 years ago. And then also more recently, the ability to use ultrasound and other imaging techniques to look at joints is a lot better than my ability to examine somebody's joints in rheumatoid arthritis. So I think we should be able to get there, but we're not there yet. And I think that there isn't going to be one test. There isn't going to be one test that goes across diseases, and for any given disease, I don't think there's going to be one test. It's going to be in combination with the ones that we have. So the SED rate and the CRP actually are useful, but we're going to have something that doesn't just say the same thing in a different way, but something that says something different about an organ being damaged or about damage to a blood vessel in particular, rather than just inflammation in general.
Okay, I think another challenge for the lay people that we're out here is understanding kind of how does this research be applied, and I think I love the enthusiasm of this question. That I know the TAPER study is basically just enrolling patients and launching, and we're asking, well, when will those results be available? So I think that's probably important in understanding how we use prednisone over time. What is the typical kind of life cycle of a research project like this from inception to treatment?
Yeah, so that's really important. I mean, once you actually start enrolling, I won't even go into the how do you get approval to start a study, but that's a study that will probably enroll fairly quickly, and we may get our target in 6 months. Some of the bigger ones, some of the larger studies with rare diseases where it's more selective, it can take a couple of years. So the AGATA study, which I mentioned, of abatacept in giant cell and Takayasu, I think it took us 3 or 4 years to enroll everybody. And then you have to follow the last patient for a period of time specified in the protocol. With TAPER, it's short, so I think we'll probably enroll people in 6 to 12 months. They'll all be followed for 6 months, but it takes months to actually adequately analyze the data, and then it takes a little while. It takes actually too long to get it published, but it even takes a while, even before you really know and honestly can say what the results are, to make sure that you're not doing something wrong. So that's going to be one of the quicker studies, and we're still talking about a couple of years.
Okay, great. I think another challenge or a question from the patient community is, what's the value or the possibility to take account what we'd kind of call alternative treatments, and we'll group several themes from the questions, whether it's an anti-inflammatory diet, whether it's different types of alternative therapies and research. Do you see that as something that's on the research agenda that's possible to do, or is it a lower priority in terms of the basic science?
Well, there are a couple of things there. I think that it's unlikely that these are going to be studied in a comprehensive way, and I think it's because these are really hard studies to do and they're expensive. No matter how you look at it, they're expensive. And I'm not just saying that in terms of pharmaceutical company development. I mean just in getting the protocols done and approved and having multiple sites up and running. That doing a high-quality clinical trial is actually is a lot of work, and you have to have kind of a good scientific hypothesis to start with. Doing that without a controlled trial is very perilous. To try to decide whether something works by going back and interviewing people and seeing whether they take supplements or not, and then seeing how those patients do. You could conceivably do such a study, but it would be very hard to interpret. I always tell patients that they're unlikely to do themselves any harm, and then the other way I kind of validate that concern is that the interaction between our diet, or what's even now referred to as the microbiome or the microbiota, all of the different microorganisms that live inside us, is massively complex, and it's increasingly regarded as important. And so there are some diseases in which there's no question that alteration in what happens in the GI tract can make a big difference in what happens in the disease. And the classic is inflammatory bowel disease, but there are others. And so it's not out of the question that those kind of environmental influences will play a difference and make a difference in our diseases, but there isn't yet any evidence that there is.
Okay, I think that's a great answer to a tough question. One final question, I think, to sum up, and if we didn't get to your specific question, we apologize. We had great participation from all of our attendees today. Lots of really good, I think, meaningful questions for the group. But there's a question here I think that's really appropriate to end on, and it's really around the idea of the fact that we have multiple vasculitides or multiple diseases. Have you found that when you do a study against one disease, you may find benefit or an insight that can be applicable to funding studies in other diseases that are similar?
Yeah, you sure can. And you hope that you'd like to have a rationale. At some point, a lot of times effective treatments are just going to be tried in other diseases. The question is on what level and at what level of funding. But it helps to have the basic science researchers looking and giving us some idea that, for example, just because rituximab works well in diseases associated with ANCA antibodies doesn't mean it's going to work in other forms of vasculitis. And if I were to predict, just based on what I know and what other people who look at things in the lab have told me, is that it is unlikely to work in Takayasu, or giant cell arteritis, because there's no convincing association of an antibody with those diseases, and because you don't see a lot of B cells hanging out in the affected arteries. Doesn't mean it's impossible. It will probably be looked at, but won't be looked at in a big clinical trial unless some small studies are very positive. Our pharmaceuticals are a little more complex than we might realize, so maybe it does work, but I wouldn't be very optimistic. On the other hand, just in terms of how polyarteritis nodosa is thought to work, it's very reasonable that rituximab would work in that disease, and so it'd be nice to at least accumulate enough data from multiple people to say maybe we should do a trial. The more rare the disease is, the harder it is to do a trial. And then, I think a drug like mepolizumab, which goes specifically after eosinophils, that's not going to be a high priority for trying in other forms of vasculitis. But so you base it on what you know about what the scientists have determined in the lab is likely to be important. If there's an animal model, that's very helpful. But you have to keep an open mind that maybe you don't understand the whole system too.
And with that, I'd like to extend my thanks to Dr. Monach for just a tremendously informative and I think exciting and encouraging presentation about what's going on in the research of these diseases. I would like to thank everyone for attending and participating. I'd encourage you to go to the Vasculitis Foundation website at www.vasculitisfoundation.org, become a member, find out more about how you can participate in the group. This webinar has been recorded and will be posted at a future date, and you'll be able to find that via social media outlets and the Vasculitis website. And I hope everyone has a great day.
Okay, thank you very much, George.